Namaskaram to all. We are discussing about genetics from Unit 18. Let's recollect the previous class. We discussed about Gregor Johann Mendel, who is an Austrian monk, discovered the basic principles of heredity through his experiments on pea plants. So he is called as father of genetics. He did monohybrid crosses involving inheritance of only one pair of contrasting characters are called monohybrid cross. For example, he crossed two forms of a single trait, like a cross between tall and dwarf plant. So he taken a single trait, that is stem height. So he taken two parents, one tall parent, one dwarf parent. In F1 generation, we get a hybrid, capital T, small t, but phenotypically all are tall. Phenotype means morphological expression, genotype means genetical expression, right? In F2 generation, we get genotypically one tall, one dwarf, two hybrids, but phenotypically we get three tall and one dwarf. Right. Let us move on to the next topic, dihybrid cross. Inheritance of two pairs of contrasting characteristics at the same time. So Mendel chosen shape and color of seeds for this dihybrid cross, that is round yellow seeds and wrinkled green seeds. Here round yellow dominant character, wrinkled green recessive character. In F1 generation we get round yellow seeds, because we know always in F1 generation dominant character will express. In F2 only recessive characters also will express, right? So by using Punnett square we can calculate, that is manipulate, that is RY, capital RY, small r, capital Y, small r, capital Y, capital R, small y, capital R, small y, small r, small y, small r, small y. So we get 9 yellow round, 3 yellow wrinkled, 3 green round and 1 green wrinkled. So at the end of dihybrid cross we get 4 types of plants, that is yellow round, yellow wrinkled, green round, green wrinkled, and we had a new combination, that is yellow wrinkled, likewise green round. Okay, next, Mendel's laws. So based on his experiments of monohybrid and dihybrid cross, Mendel proposed 3 important laws. They are called Mendel's laws of heredity. First law, law of dominance. So capital T, small t, that's the hybrid we get through monohybrid cross. In F1 generation we get capital T, small t, it's a hybrid, but phenotypically it's tall only, because it has capital T and small t. So capital T is the dominant character, always expressive. Small t is the recessive character, always suppressive. Next law, law of segregation. Actually genes are in the form of pair, that is for example இப்போ capital T small t, இப்போ கேமெட் ஃபார்ம் ஆகும்போது அந்த கேபிட்டல் டி ஸ்மால் டீயும் ரெண்டாக பிரிஞ்சிடுது, கேபிட்டல் டி தனியாக பிரிஞ்சிடுது, ஸ்மால் டி தனியாக பிரியுது. இதுதான் செக்ரிகேஷன். ஸோ வென் கேமெட்ஸ் ஆர் ஃபார்ம்ட் த allelic pair get separated. ஸோ இதுதான் லா ஆஃப் செக்ரிகேஷன் பார்க்குறோம். நெக்ஸ்ட் லா ஆஃப் இண்டிபெண்ட் அசார்ட்மெண்ட். இப்போது எல்லோ ரவுண்டு அப்படின்றது ஃபார் எக்ஸாம்பிள் கேபிட்டல் ஆர், கேபிட்டல் ஒய், இது ஒரு டைஹைப்ரிட் கிராஸில் பார்த்தோம் இல்லையா. இதுதான் வந்து பியூர் பேரண்ட். தட் இது எப்பவுமே ஒன்றாவே இருக்காது. இந்த கேரக்டர்ஸ் மாறலாம். ஃபார் எக்ஸாம்பிள் எல்லோ ரிங்கிள்டு அப்படி மாறலாம். அந்த மாதிரி க்ரீன் ரிங்கிள்டு தான் இருக்கணும்னு இல்லை, க்ரீன் ரவுண்டாக கூட மாறலாம். ஸோ இண்டிபெண்ட் அசார்ட்மெண்ட்றது இந்த ஃபேக்டர்ஸ் எப்பவுமே ஒன்றாவே இருக்காது, எப்படி வேணாலும் மாறலாம். ஃபார் எக்ஸாம்பிள் எல்லோ ரவுண்ட் அப்படின்றது எப்பவுமே கண்டினியூ ஆகாது, எல்லோ ரிங்கிள்டு ஆகலாம். அந்தமாரி க்ரீன் ரிங்கிள்டு எப்பவுமே கண்டினியூ ஆகாது, க்ரீன் ரவுண்டாகவும் மாறலாம். நெக்ஸ்ட் டாபிக், குரோமோசோம்ஸ், DNA and genes. The human body is made up of million cells. The nucleus of each cell contains thin thread-like structures called chromosomes. The term chromosomes was first coined by Waldeyer. Chromosomes are the carrier of genetic material which contain the hereditary information. The chromosomes are highly condensed coil chromatin fibers packed with the DNA that forms the genetic material. Genes are nothing but the segments of DNA which are responsible for the inheritance. Next, structure of chromosome. Chromosomes are again thin, long and thread-like structures consisting of two identical strands called sister chromatids. They are held together by the centromere. Each chromatid is made up of spirally coiled thin structure called chromonema. Chromonema has a number of chromomeres. The chromosomes are made up of DNA, RNA, chromosomal proteins, that is histones and non-histone proteins, and some metallic ions. So chromosome consists of the following regions: primary constriction, secondary constriction, telomere, satellite. Primary constriction, the two arms of a chromosome meet at a point called primary constriction, that is centromere. Secondary constriction, other than centromere, wherever the constriction takes place, that they are called secondary constrictions. One is happened in the telomere region, another one is happened in the satellite region, so they are called secondary constriction. Telomere is present at the one end. It maintains and provides stability to the chromosomes. Satellite, a knob-like structure. If satellite is present, that chromosome is called SAT chromosome. Next, types of chromosomes based on the position of centromere. So based on the position of centromere, the chromosomes are classified as telocentric, acrocentric, submetacentric, metacentric. Telocentric, the centromere is found on the proximal end. They are rod-shaped chromosomes, so only Q chromosome is present, P chromosome absent. Acrocentric, the centromere is found at one end with a short arm and a long arm, so this is also a rod-shaped chromosome. Submetacentric, it has two unequal arms, L-shaped. Metacentric, the centromere occurs in the center of the chromosome and forms two equal arms, so it is V-shaped chromosome. Next, types of chromosome based on function. Eukaryotic chromosomes are classified into autosomes and allosomes. Autosomes means body chromosomes, they determine the body characters. Allosomes are called sex chromosomes, they determine the sex of the individual. If it is XY, it's male. If it is XX, it is female. Next, karyotype. Karyotype is the number, size, and shape of chromosomes in the cell nucleus of an organism. Ideogram is the diagrammatic representation of karyotype of a species. It consists of all the metaphasic chromosomes arranged in homologous pairs according to decreasing length, thickness, position of centromere, shape, etc., with the sex chromosomes placed at the end. In the body cells of sexually reproducing organisms, chromosomes generally occur in pairs, so they are called diploid, 2n. When they form gametes, the chromosome becomes single set, so it is said to be haploid, n. So let us take human chromosomes. There are 23 pairs. Out of 23, 22 pairs are called autosomes, body chromosomes, and one pair, that is 23rd pair, is called allosome, sex chromosome.